All right, well, one person who's physically here had to go run and grab something from their car. So I guess the first thing I'll do is give you the attendance word for today. The first one is going to be from one from yesterday, since yesterday was the first day of fall. Today's the first full day of fall, because technically fall didn't start until like 9 o'clock or something like that. So the first word for attendance today is fall. And as usual, I'll remind you, send in those keywords before 9 a.m. today, because if you miss the 9 a.m. cutoff, then you just have to do the questions when I post them. Well, like I said, I wanted to get a slow start because I don't want her to miss anything. But let me ask you this, which is only one person here, but this goes for somebody online too. Does anybody know how much independent work is worth? So if you write a page, how much is a perfectly written page worth? About how many points? Everybody knows?
14.
Here, okay. I see somebody answered, I just can't hear you for some reason.
14.
Yeah, I see you're talking because like your little microphone thing flashes. But there's an issue on my end and I can't hear you. So you probably guessed it, but 4 is wrong. Good guesses though. There we go. You're a little bit closer. So somebody here kind of guessed correctly and said 10. Now, if you remember back when I first told you about independent work, and if you read the syllabus like I told you to, it says independent work is worth 10 points per page, right? That's what it says in the syllabus. That's what I told you to begin with. But had you been reading these announcements that I keep begging you to read, you would see that they're actually worth more than that right now, right? A few weeks ago, I put out an announcement that said, hey, by the way, if you do your independent work now, it's worth 15 points per page. And it's going to keep going down, so the sooner you do it, the better. And right now, it is down to 12.25 points per page. The point here is, read the announcements. You guys are missing out on 3 points because you're not reading the announcements and you haven't done your independent work, and you would have a lot more points had you done it. Another way I know that you guys aren't reading the announcements is this right here. So many of my announcements have this. I know it's a little bit hard to read. This one says, speaking of which, reply to this if you read it for half a point of independent work. So had you guys been reading my announcements and replying to them and saying, hey, I read that, you guys have been stacking up the independent work points just from doing that. So please read the announcements. And apparently some of you have read none of them, so let me put it this way. If you look really closely, I know they're really long, but from here to here, that's the new information. There's not much. I'm not giving you new information, or I'm not giving you much new information. It's a very small amount of information. And then you read right here, it says old information. You aren't required to read this, right? So I get it if you don't want to read that whole long thing, but this short information is important. Please read it. You guys are missing out on points. Any questions? Well, shoot. Yeah, any questions about that? If you guys are online and have questions, I guess you'll have to type because I can't, apparently my speakers aren't working. I can't hear you. All right, so anyway, now that I've reminded you of that, let's talk about this really quickly. I'll remind you that pre-lab corrections are due on Sunday. So the last pre-lab you did, that's the same pre-lab for this coming week. So if you got a perfect 5 points, there's nothing to do. Just relax, enjoy your weekend. But if you have room for improvement, go ahead and make your corrections so you can get full credit. Or if you don't make any corrections, whatever grade you got last week will be the same for this week. That's up to you. So any questions about that? No? Okay, this is good. Today, this is new. Office hours, I'm going to have to they're going to have to be online today, 100% online. That being said, if for some reason one of you had planned on seeing me physically in person, go ahead and email me and let me know. Say, hey, I planned on coming to see you today, in which case I'll change my plans and stay in my office and meet you. But right now, my plan is to do it 100% online. How do you meet me online? The same way you're doing it right now for those of you online. Just go here, and that's where I'll be. Red Friday, we got one person in red. If you want to take a picture afterwards, extra points. If you want to take the picture afterwards, extra points. If you're online and you want to participate in this, send me an email, we'll figure it out. Here's another thing I'm going to change. If you ever meet me online once a day for starting next week, if you meet me for office hours, I'm going to give you points for independent work just for meeting me. I don't know how many yet. That's why it's not going to start until next Monday. I'm going to think about how much it's going to be worth. And all I want to do is just meet you for like 5 minutes. If you have any questions, I'll answer them, but more than likely, you might not have any questions. So I just want to talk to you about your performance and say, look, you're doing good. You're doing a really good job of this. Keep that up. You might want to do a little bit more independent work, or let me tell you how you can do better with your pre-labs, whatever it is. I just want to meet with you for 5 minutes and kind of nudge you in the right direction because we're almost halfway through the semester. So it's time to really start nudging you in the right direction. Any questions about that? All you have to do is meet me. And some of you are doing really good, and all I'll do is say, hey, good to talk to you. You got your extra points. See you later. Right? There's not going to be much to say. Speaking of which, some people really need work with this, independent work. And I'm not talking bad. I know this is new to a lot of you guys, but for your independent work, you really need to write it in your own words. So if you're going to summarize an article, literally read the article as many times as you need to, put it away, and then just write from memory what you remember. A lot of you, or some of you are doing, what you're doing is basically taking the thing and just rewording it. So it's like, endangered species are endangered because of habitat loss and genetic loss. And then you might change that to say, endangered species are endangered because of genetic loss and habitat loss. Like you just swap around those two words. That's not working. That's technically plagiarism. But I'm not getting on to anybody for it because you just might not be used to having to write in this way. And if you are one of those people I'm talking about, I'm actually commending you because lucky for you, you've been doing independent work. So you're starting to learn what you need to do. Some people are going to wait until the last week of class to start writing papers. And then I'm going to send them those same things that I've been sending you, and they're just going to be out of luck. But you who are learning those lessons now, that's good. Now you got time to fix it and adjust yourself. But anyway, any questions about independent work? And again, there's a couple of you probably know who you are. I'm really not getting on to you, like talking bad about you for what you're doing for independent work. Good on you for getting it done early. We're going to learn these lessons together before it's too late. So, yep, that's it. Anything else? Any questions? Concerns, questions? All right, say questions. All right, for attendance, I just want to make sure I've got everybody. Hold on, let me fix this. Where are we at? Lost my mouse. There we go. All right, really quickly, I'm going to go through attendance. I'm just going to call out your name if you're not here. So obviously, if you hear your name, let me know that you're here. And by here, I mean either online or in person. And if you're online, I guess you're going to type it because, like I said, I can't hear you. Anyway, Sims, Fisher, Strickland, Whitaker, Stanley, Newton, Mark, Smith, Carr, and Fawcett. That's who I have that are not here. All right, guess we're good. Now then, let's jump back into chapter 6. Hopefully we can finish it today. The main stuff that we talk about in chapter 6 is cellular respiration, right? The glycolysis, the citric acid cycle, the electron transport chain. That's the big part we talked about. Before we start talking about that, I gave you a little bit of a brief introduction, an overview of photosynthesis and respiration. That's not that important as far as the exam is concerned. After I'm done talking about this, we're going to talk a little bit about fermentation. That's going to be very quick. And there's not going to be much about it on the exam, so that's not very important. We're talking about the very important stuff right now. So before we finish up the last stage of cellular respiration, which is the electron transport chain, let me remind you what we learned, the important stuff, the big stuff about the first two stages. First of all, the first stage is called glycolysis. It is the one and only stage that does not happen in the mitochondria. You need to know that for the exam. All the other ones happen in the mitochondria. That's the only one that happens out of the mitochondria. What is the input of glycolysis? Glucose. Or more specifically, the way the question is going to be asked on the exam is, which of the three stages directly uses glucose? And the answer is glycolysis. So anyway, glucose goes into glycolysis, pyruvate comes out. So there's another test question. Which of the three stages produces pyruvate? Glycolysis. In the process of breaking down that glucose to pyruvate, we do make a little bit of ATP. So a test question, which of the three stages make ATP? All of them. Anyway, so yeah, we make a little bit of ATP, but more importantly, what we're doing when we break down this glucose to pyruvate is we're stripping away these high energy electrons because those are all going to go to the electron transport chain, which is what we're going to talk about once I'm done catching you up. So that's it for glucose. I mean, that's the big picture. There's other little details we've already talked about. We also mentioned that pyruvate cannot go into the mitochondria, into the citric acid cycle. So we had to do a little bit of changes to it. We knocked off a carbon and made acetic acid. That's what that AA stands for. And then that acetic acid had to be attached to an enzyme called coenzyme A, and that gave us acetyl-CoA. blah, 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 blah. The important thing here is, as we were doing that, we were producing carbon dioxide. So, test question. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages is associated with acetic acid? Citric acid cycle. Which of the 3 stages is associated with acetyl-CoA? Citric acid cycle. And again, most importantly, even though we did make a little bit of ATP and the byproduct was carbon dioxide, most importantly, what we did here in the citric acid cycle was strip away high energy electrons, just like we did in the first stage, because all those high energy electrons are going right here to the electron transport chain, which is what we are about to talk about. And then remember, the electron transport chain is the one that produces the most ATP. And if you can remember that one fact, and hopefully that'll help you remember that the other 2 were just there to support the third one. For those of you who did the video from the last lecture, a lot of people got that question wrong. How did I word it? Well, anyway, what you need to know is high energy electrons are stripped away in the first 2 stages, and then they're used in the 3rd stage. So usually the questions on the attendance videos aren't important. They're usually just to make sure you're paying attention. But that was an important question. So make sure you know that the first 2 stages strips away high energy electrons, and the 3rd stage uses them. And that's what we're going to talk about, is how they are used. If I can get this working. Come on. Bear with me, I apologize. There we go. Now we're cooking. So we already talked about this. I already said this. This is kind of how we left off on Wednesday. We were talking about how it's a downward trip. I keep using the word high energy electrons. By the time this is all done, they're just regular old electrons. And in the process, they're giving off their electricity, their energy. So here we go. There's an overview of the whole thing. This is an overview of the entire cellular respiration if you're focusing on the electrons. So if you're focusing on the electrons, and here's the test question, where do the electrons come from? They come from food. They come from the glucose. Food's a better word for it because, we don't always just eat glucose, but I always say glucose. Those high energy electrons come from glucose. They're stripped away as the glucose is broken down into pyruvate, which is then broken down into acetic acid, which is then broken down into the citric acid cycle, right? We keep stripping away these high energy electrons. That's what that NADH is, and also the FADH2. Remember, that could also be FADH2. You don't need to know the difference between them. Like I've said many times, anytime you see anything that looks like that or this, you should think, yep, that thing's carrying electrons. Anyway, we're stripping away high energy electrons from the food. And as they go through these steps, down, 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 they're putting off their energy because they are high energy. And as they put off that energy is then used to make ATP, which is the focus of cellular respiration. It's all about making ATP. And these little three dots is what we're about to talk about. Because right here it just says, okay, they're going from step to step and they keep putting off energy. I'm going to explain how that happens here in a second. But like I said on Friday, when we ended things, electrons have to come from somewhere. They have to go somewhere. Where do they come from? They come from the food. Where do they go? Oxygen. Oxygen is the thing, the final acceptor of the electrons. It is the thing that takes those electrons. That is where the electrons end up. Now, granted, when that happens, that also produces a molecule of water. But the answer is, and that will definitely be a test question, where do the electrons go in cellular respiration? And the answer is oxygen. And this one will be a little bit tricky because one of the options is going to be NADH or FADH2. And don't let that trick you because NADH or NAD+, whatever, it is the thing that accepts the electrons. That's true. But that's just to move them around. The final resting place of the electrons, excuse me, is the oxygen. Any questions about this slide? And again, I'm going to go in more detail. This is just a big overview of what we're about to talk about. All right, I think I also mentioned this earlier, but the proteins of the electron transport chain are built into the inner membrane of the mitochondria. You'll see in the next page that the mitochondria has 2 membranes. So think about that for a second. I know we didn't do chapter 4, so you might not know this, but our cells are one membrane, right? We have one, it's a bilayered membrane, but we just have one membrane in our cell. These things have 2 membranes, and that's going to be important here in a second. You'll see why. Also, the inner membranes are really highly folded, and again, that's going to be important here in a second. Not that I'm going to test you on it, but this will help you remember it. Why is it highly folded? It's going to give you a large surface area, therefore more copies of the electron transport chain proteins. You'll see what I mean when I show you the picture. And this is just a really great example of how biological structure fits its function. So I'm not going to take questions yet. Let me repeat this, but show you the picture of what I'm talking about. So here's the mitochondria. Like I said, it has two membranes. Here's the inner membrane that we can see. That would be like us. That would be like our cells, just the one membrane. But then imagine our cells had an extra membrane on the outside. That's what these mitochondria have. So they have an inner membrane and they have an outer membrane. That inner membrane is really big. You can see it's all crinkled up, right? So imagine, is there a garbage can in here? There used to be. There used to be a small one. Oh well. Imagine taking like a big old 50-gallon garbage bag, right? And having like every square inch have one protein. Obviously it wouldn't be a protein, but imagine you could. One protein on every square inch, right? Of a bag, whether it's a big bag or a little bag. Obviously a big bag, if there's one protein for every square inch, it's gonna have more proteins on it, right? Because you have a big bag as opposed to a little Kroger bag. That's what's going on here. So you have this huge big old bag that has a bunch of proteins on it, and then you want to fit it into a Kroger bag, a smaller bag, so you crumble it all up and it's all wrinkly. That's kind of like this, right? So it's really big in there, and the reason it's really big is because the more surface area you have, the more proteins you have. And it's the proteins that are doing the electron transport chain. All that information I just told you will not be on the exam. That is just so you understand how this process is going to work. Because if you understand how the process works, then you can remember the few details that I will ask you on the exam. I will tell you this, I'm going to put a big X through this, because what I'm not going to do is show you a picture of the mitochondria and ask you to label the parts. So you don't need to remember, you don't need to study that. And another thing, I'm not even going to use these words, so I'm going to put an X through them. The space between the membranes, now that is important, and you'll see why. That's going to come back later. So sometimes things get stuck between the inner membrane and the outer membrane, and that's a really important fact, and you'll see why here in a second. And who here knows how a hydroelectric dam works basically? I mean, it's right there. But. So basically what happens, if you can see this picture, on the other side of this dam that you can't see, right, in that direction, is like a lake, right? A big body of water. And that dam is holding all that water back, and that water wants to come this way. So there's a lot of energy, a lot of potential energy there, right? It's pushing. Gravity is making that water push the dam. Of course, the dam's not moving if it's built properly, but that water wants to rush that way. So what happens is, as you can see here, there are little areas where water can flow, right? It lets the water through the dam. Then the water's coming through really quickly because that's just what nature does, right? Gravity's making it go downhill. And from that kinetic energy, right? We had all that potential energy because the water wants to run downhill. Then it does run downhill. That's kinetic energy. That turns a turbine, and that produces electricity, right? That's how a hydroelectric dam works. Now, it's very similar to how the mitochondria works. Let's talk about why. The energy stored by the electron transport is something like the water behind a dam. What happens is we get these hydrogen ions that gush back towards where they're less concentrated. And this is really hard to see, but again, there's the inner membrane right here and the outer membrane right here. Let me zoom into this part right here and try to draw what we see here. So there's the inner membrane, here's the outer membrane, right? This is a bunch of stuff on the inside. And then this is the space in between the membranes. So what we have is all these hydrogen ions that are sitting here. There's a lot of hydrogen ions inside or in between the membranes, but there's not very many hydrogen ions inside, right? So here there's a bunch of hydrogen ions, here there's not. So you know from chapter 5, things want to diffuse. Those hydrogen ions want to go down. They want to go from this area of high concentration to the area of low concentration. And that's how it works. And what happens is when they do, there's a protein that lets them through, sort of like these holes in the dam, and there's literally a turbine on those proteins, and it turns, it really does turn, and that's what produces the ATP. So we have all these hydrogen ions built up, and they just go down, and as they go down, as they diffuse down, that's facilitated diffusion, they turn, and that makes the ATP. So really the only question is, where did we get all these hydrogen ions? Why do we have such a high concentration of hydrogen ions in there? And that's what we're going to talk about as we move forward. Here's something you need to know though. Like I said, the hydrogen ions go from that inner membrane, that in between, the inner space in between the membranes, and they diffuse down. So then the question is, what is that protein that lets them through and that makes the ATP? And you need to know this for the exam. It's called ATP synthase. It's the air. It feels like it's 78 in here. So yeah, know that word, ATP synthase. Oh, yeah, it's 80. I wasn't too far off. This is what happens as soon as we get cold days. People cranking it up all the way. Anyway, yes, the enzyme that makes ATP in the electron transport chain is called ATP synthase. Please don't mess that up. It's in the name. Like I told you in chapter 5, enzymes are named after what they do, right? In this case, it synthesizes ATP. And enzymes end with the last 3 letters ACE. So it's in the name. The enzyme that makes ATP in the electron transport chain is called ATP synthase. Any questions about that? The next slide I'm going to show you is basically a picture of everything I've just told you in the past 2 slides. The next word for attendance, speaking of which, be this one. I'll circle it. So again, if you're listening to the lecture, you've got your eyes closed. There's nothing wrong with that. But open your eyes for a second and read this word. The one that I have circled and the one that I'm drawing an arrow to is the second word for attendance. Anyway, any questions about what I've told you so far? And again, I'm about to explain it again with a picture. All right, this picture is much better than the one I tried to draw. Here we go.
What's the word?
Let me get a mitochondria. Let me do it like this so they can see.
I can't see what you circled.
Looking at the
It's
mitochondria,
slide, man. Just slide.
right here, right? There's a big mitochondria. Then we're going to zoom
It's
in.
slide, S-L-I-D-E.
So, as we zoom in, you can see that's the inner membrane, right? That's the inner membrane. That's the outer membrane. So this is the space in between, right? And we have proteins embedded in this inner membrane. So again, to remind you, oh yeah, right there, space between membranes. So what's happening is all these high energy electrons that we strip away from the first 2 stages, from glycolysis and the citric acid cycle, we're stripping away these high energy electrons using this NADH and also FADH2, whatever, it doesn't matter. They come to the electron transport chain and literally jump from protein to protein. Again, these proteins are embedded in the membrane. Just like in chapter 5, when we talked about proteins embedded in the membranes, here we go again. Now we're talking about them, and they actually have an important purpose, at least these do. And just like in chapter 5, we talked about not facilitated diffusion, which we did talk about that, but active transport. Remember, active transport uses proteins to pump things against their concentration gradient. That's exactly what's happening here. These are proteins with little holes in them, and they're literally pumping hydrogen ions from an area of low concentration to high concentration, right? That's the opposite of diffusion. It takes energy to do that. It's like pumping water, right? Pumping water back up the dam, so to speak. So that takes energy. And in this case, that energy is coming from those high energy electrons that we strip away. So again, it just goes protein to protein. Each step it loses a little bit of energy. That energy is released. That energy is used to pump hydrogen ions against the concentration gradient. So now what do we have? We have an area of high concentration of protons and low concentration. And as you know, again from chapter 5, this naturally just wants to diffuse back down. But it can't go directly through the membrane. So as you learned in chapter 5, there is something called facilitated diffusion, which I use the analogy of if you had 500 people in this room, you know, and there's nobody in the hallway, people are going to want to diffuse out, right? But they can't. People can't just squeeze out through that hole, right? So what would I have to do? I would have to open the door. And the door would then be a protein, right? The protein that allowed it to happen. That's what this is. That's what ATP synthase is. It's a protein that allows these hydrogen ions to flow back down, to diffuse back down to the area of low concentration. And as they flow through, it literally spins that protein. And that's where we make ATP. And that's it. That's how it works. So that's why those first 2 stages, that's why I kept saying the first 2 stages are all about supporting the 3rd stage. Because remember, and again, this is going to be a test question, which of the 3 stages produces the most ATP? It's this one. So the first 2 are all about supporting the 3rd one. And that's how it works. We need to pump hydrogen ions against the concentration gradient so that they can flow back down the concentration gradient. Where do we get the energy to pump those hydrogen ions? Well, we get them from all the high energy electrons that we stripped away when we broke glucose down to pyruvate, and when we broke pyruvate down to acetic acid. And when acetic acid went in and joined that 4-carbon molecule in the citric acid cycle and became citric acid, and we broke that down, so on and so forth. Yeah, so that's it. You'll notice there's a little skull and crossbones here. I mentioned this earlier. But the reason that that's there is because, again, if you ever wondered what exactly is it about not breathing that kills you, that's what it is. Because if you don't have oxygen here at the very end, it's like not plugging something in. There might, like if I have something, let's see, I don't even see a plug. There's a plug. There's electrons ready to come out of there right now, right? They're just sitting there. They've been pumped from John Amos. They're ready to go, but they're not going anywhere because there's nowhere to go. If I were to plug something up, then the electrons would flow, right? It's the same here. You have all these electrons, but if there's no oxygen, if there's nowhere to go, they're not going to flow. If the electrons aren't flowing from protein to protein to protein, then you're not pumping your hydrogen ions. Therefore, hydrogen ions aren't diffusing back down their concentration gradient. Therefore, you're not making ATP. Therefore, your energy or your cells can't do anything because they don't have any ATP energy, and they die, and therefore you die. All right, any questions about that? I will put some nexus through this to help you with your studying. Notice the fact that there's 3 proteins. That's actually not on accident. There really are 3 proteins for each step, so you don't need to know that, so I'm going to put an X through that. Notice the fact that NADH is the one that brings electrons to the first protein, and FADH2 is the one that brings it to the second protein, but you don't need to know that either. I'll put an X through that. I keep saying that oxygen is the acceptor molecule. You do need to know that. And you do need to know that once the oxygen accepts those electrons, then you get yourself a molecule of water. You do need to know that. Obviously, oxygen and an electron don't equal water, right? So it should be common sense that yes, you also need some hydrogens, right? Because water is H2O. But despite that, I'm going to put it next to that because you don't necessarily need to know that. I'm not going to ask you how many oxygen molecules combine with how many electrons to make how many molecules of water. You don't even know that. Just know that the electrons eventually are accepted by oxygen, and that forms a molecule of water. And here's a little preview, a little hint for you. When we talk about photosynthesis, once you memorize this, photosynthesis should be kind of easy because it's literally the opposite. So in photosynthesis, water is like, all right, you're going to need some electrons, so here's some regular old electrons. And water donates the electrons, which produces oxygen. And then photosynthesis energizes the electrons, right? So here the electrons lose their energy and get accepted by water, and get accepted by oxygen, which produces water. And photosynthesis is the opposite, so just something to keep in mind. We'll get there next week when we talk about photosynthesis. Any questions about this slide? All right. Here we go. We can make it through this before the day is over. The results of cellular respiration. As I've already told you, you need to know this number, and it varies depending on your source. But we say in this class, according to this textbook, that we generate about 32 ATP per molecule of glucose. And that is the number you need to know. And like I said on Wednesday, I'm not going to be tricky, right? So like I just now said, other sources say other things. That video I showed you, which you should watch if you haven't, with Hank in it, he says 38 ATP. So certainly one of the options on the multiple choice exam is not going to be 38. I think that would just be tricky. That would be fair. Because again, if you're reading 38 in one source and 32 in another, that's tricky. So most likely your options on this multiple choice exam are going to be, how many ATP are made? 32, 42, 52, 62, something like that. It won't be tricky. If you can remember about 32, you should be okay. Here's a good test question. Not test question. A good independent work topic if you want to look into it. If cellular respiration requires ATP, this is a good time for me to remind you that glycolysis had the energy investment phase and the energy harvest phase. So you had to start the whole thing off with a little bit of ATP. The very first step requires you to put some ATP in. If cellular respiration requires ATP, but cellular respiration is the thing that makes ATP, then where did the first ATP come from? You can look that up for independent work if you want. It's trivial. You don't need to know it for this course, but this is something you might look into. Again, this is an overview of everything we've already learned, so I'm going to say this again, but very, very quickly. First of all, I'm going to get rid of that word acid, call it pyruvate. Anyway, all the test questions, right? Which of the 3 stages happens outside of the mitochondria? Glycolysis. Which of the 3 stages produces ATP? All of them. Which of the 3 stages produces the most ATP? Electron transport chain. Which of the 3 stages happens inside the mitochondria? Citric acid cycle and electron transport chain. Which of the 3 stages uses glycolysis? Glucose. Glycolysis. Which of the 3 stages produces pyruvate? Glycolysis. Which of the 3 stages produces carbon dioxide? And that's not shown on here, but citric acid cycle. Which of the 3 stages strips away high energy electrons? Glycolysis and citric acid cycle. Which of the 3 stages uses those high energy electrons that have been stripped away? Electron transport chain. All right, so that's it. I mean, there's other test questions, but that's the big part. I'm going to put it next to the 2, because remember, you don't need to know the exact number. You just need to know that the first 2 produce a little bit of ATP, and the third one produces a lot, about 32. All right, so that's the overview. Any questions about that? All right, I asked this on Wednesday. I'll ask it again. Who here has had breakfast? Have either of you had breakfast yet? Okay, so you guys ate. Did you both eat pure glucose? No, or no, right? Probably not. What did you have for breakfast?
Pop-Tart.
Pop-Tart, see? There's probably more fructose and probably some sucrose, but I doubt there's any glucose by itself in it, right? So she didn't have that. What did you have for breakfast? That's what I had. Oatmeal and bananas and walnuts. So point is, all the 3 people in this room ate, but we didn't eat glucose. And everything I keep talking about is glucose, 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 glucose. But really quickly, just to acknowledge this, not that I'm not going to test you on it, but it is worth mentioning. Obviously, we can digest other stuff than glucose, right? Now, granted, if she had sucrose, which she did, that is probably broken down into glucose, which was that. But your book does point out that we do eat other stuff, right? So it's not like it's all glucose. As you know from chapter 3, there's nucleic acids and carbs and fats and proteins. And depending on what it is, it's broken down and it starts at different levels. For example, if you eat proteins, as you already know from chapter 3, the monomer of proteins are amino acids. So you break those down into their individual amino acids. And remember, there's 20 different of them. So depending on which one it is, the amino acids themselves are broken down. And depending on the parts that you use, that might go into glycolysis if it's a sugar. It might go into this portion where you're turning pyruvate into acidic acid, or it might go directly into the citric acid cycle. Fats, again, we know that like triglycerides, that's the kind of fats we're talking about, that's broken down into glycerol and 3 fatty acids, right? So you break that down, the glycerol can go directly into glycolysis. The fatty acids can get groomed and then go into citric acid cycle. So I'm going to put a big X to this. You do not need to know this for the exam. Now, who knows? Maybe I'll have an extra credit question. Probably not, but that would be a good extra credit question. Like if you have proteins, which of the 3 stages would they go in? The answer would be all 3. If you eat fats, which of the 3 stages would they go in? And the answer would be tricky, because this is going to go directly into the citric acid cycle. But anyway, I'm going to move forward. I'm not even going to take any questions. If you have questions, maybe during office hours, that won't be on the exam. And we're running, not short on time, but almost. So that's it for cellular respiration. That is it for the most important part of the exam. That middle bullet point that we just finished is going to be the bulk of the exam as far as this chapter is concerned. There are going to be a few questions about what I'm about to ask you, but not many. So I keep saying oxygen is that final acceptor of the electrons, right? I keep saying that. Electrons have to come from somewhere. They have to go somewhere. Where do they come from? They come from glucose. Where do they go? They go to oxygen. Well, what happens if you don't have oxygen? Like I said, you die, right? How many of you ever held your breath, though? Have you ever held your breath? Did you die? No, she's still alive. Have you ever held your breath? Yeah, you're still alive. I've held my breath. I'm still alive. So obviously, when you don't have oxygen, it doesn't kill you right away. So how does that work? How are you still alive if you didn't have oxygen? Well, cells can work a short period without oxygen. And when that happens, that is called fermentation. And you need to know what fermentation is. Fermentation is the anaerobic harvest of food energy. So we're still talking about breaking down glucose or yeah, we're going to talk about glucose, but again, keep in the back of your mind that it's actually in food. We're still breaking down the food. We're still stripping away high energy electrons, but we don't have any oxygen to accept it. This is a process that's anaerobic. It's without oxygen, so it makes it trickier. And that meme is an old one at this point. Also, to remind you guys, again, I don't make these memes. I don't even go looking for them. I just find them. And when I find them, I harvest them. Anyway, there's all kinds of different fermentation, and we're going to look at two different types. We're going to look at fermentation in human muscle cells because we're human, right? So that's relevant to you. And then we're going to look into fermentation of microorganisms, and you'll see why. So how many of you have ever sprinted or done some really hard workout where you're out of breath and it starts burning? You ever done that? You've been out of breath and you feel the burn either in your muscles or in your lungs or whatever? Well, that's what this is. That's fermentation in human muscle cells. After functioning anaerobically, again, without oxygen, for about 15 seconds, muscle cells generate ATP by the process called fermentation. I'm going to put it next to this 15 seconds because that's not a number you need to know. Also, I'm sure that varies. That's part of being healthy. Like when someone does a lot of cardio exercise, that varies. Some people are better at absorbing oxygen and using it. But yeah, point being is, after you run out of oxygen for a little bit, you start using fermentation. And obviously that doesn't mean you're holding your breath. That could mean, again, like you're sprinting, you're running really fast. It's not like you're holding your breath, but your muscles are using so much oxygen that you're using more than you're taking in. So at that point, you are without oxygen. Even though you're breathing it in, you're still using more than you're breathing in. Therefore, you are technically without oxygen. Here's a good question. Again, I told you there's two different types of fermentation we talk about, but regardless of which one we talk about, fermentation relies on glycolysis. And if you watch that short little video that I shared with you with Hank, he tells you that too. Fermentation relies on glycolysis. Glycolysis is that first stage. It is the one that does not require oxygen necessarily. I mean, it helps because what it does require, remember, is all those NAD+, right? That's the thing that accepts the electrons, or technically, once it gets the electrons, technically we call it NADH. And this is a good time for me to remind you that I don't ask you to differentiate between the two. But anyway, it requires those things to strip away the high energy electrons because that's what we need, right? But again, those high energy electrons can't take those electrons anywhere because there's no oxygen, because you're out of breath. Also, I'll remind you, again, you don't need to know that glycolysis produces 2 ATP. Just remember it produces a little bit of ATP. The point here in this context is, again, fermentation uses glycolysis because glycolysis doesn't technically use oxygen. But also remember, glycolysis doesn't make that much ATP, which is why you can do that for a little bit, right? This is why you can go out of breath and have that burn for a little bit, but eventually you would die without oxygen, right? That's why. You are producing a little bit of ATP. You can sustain you for a little bit, but not a lot. All right, as I already said right here, the NAD+ has to be present to receive the electrons, right? We need, if we're stripping away electrons, we need the thing to take those electrons. And again, that's not a problem when we're talking about what we were talking about earlier. If you had aerobic conditions, this would be regular oscillative respiration. The NAD+ gather the electrons, taken the electron transport chain, they would go down the electron transport chain because the oxygen was there to catch them, but now we don't have oxygen. So here's the problem. Those NADHs, NAD+, when they get those electrons, they have nowhere to put them, but they need to put them somewhere because they need to be open again to get more electrons. So what happens is the NAD+, well, I'll come back. I'll tell you what happens in a second. So this bullet point is basically everything I just said, right? We have all these NAD+, they gather the electrons, and they have nowhere to put the electrons, therefore they can't gather any more electrons, right? And that's not a good thing. The next word for attendance, let's call it down. Word I just circled, down, is the next word for attendance. Anyway, this next picture I'm going to show you is basically a picture of what I'm saying here, and a picture you've already seen before. So we've stripped this NAD+, strips away these electrons, right, and then it's NADH. And in a normal circumstance, it would just drop those electrons off of the electron transport chain. And once it drops those electrons off, it's a bucket holding electrons basically, right? Once it dumps the electrons, the bucket is empty again. So it can go back and it can strip away more electrons. But this doesn't happen, right? Because we don't have oxygen. So this bucket of electrons cannot dump the electrons out. So what happens? Well, NAD plus, again, it can't dump them, so it has to put them somewhere. So the electrons are added to the pyruvate instead. And to remind you, with glycolysis, we start with glucose, and we break it in half to 2 molecules of pyruvate. Right? So we already know I'll just abbreviate that. We already know that the reactant is glucose. Oh, come on. And the product is pyruvate. We already know that. So now what we're saying is, because those electrons can't go to the oxygen, your cells basically make use of the pyruvate. Like, all right, well, if oxygen can't take these electrons, pyruvate, you take them. I don't need them. I need to be an empty bucket so I can gather more electrons, right? So that's what happens. And it just so happens, and this is the part of all the things I told you, I finally got the part that you need to know for the exam. What happens when you add the electrons to the pyruvate? You produce lactic acid. So the test question, remember I told you there's 2 different types of fermentation that we're going to talk about. So one of the test questions is going to say something like, fermentation in muscle cells produces what? The answer is lactic acid. The other question is going to say something like, fermentation of microorganisms produces what? And that's what we're about to talk about. And the answer is alcohol. So the only true answers when I ask you any question about this are lactic acid or alcohol. But it's a multiple choice exam, so there's going to be a bunch of other nonsense. And if you choose the other nonsense, you're going to lose even more points. So at least when you get to this, either choose a lactic acid or alcohol. At least if I give you this question and you say alcohol, at least you'll get half credit. Because at least alcohol is one of the things that fermentation produces. Anyway, again, I gave you all that detail and told you how it works. But for the exam, all you need to know is that fermentation in human muscle cells, or really any muscle cells, produces lactic acid. And like Hank said in that video, that short video I shared it with you, if that didn't happen, like if you made ethanol instead, alcohol, then working out would just get you drunk, which would be really weird. Anyway, here's a picture of everything we just said, right? Again, the input of glycolysis is glucose. We break it in half and make 2 molecules of pyruvate, right? We're stripping away high energy electrons that used to go to the oxygen, but now there's no oxygen, so there's nowhere to put those electrons. So instead, the NADH dumps those electrons back onto the pyruvate, and that gives you lactic acid. And that's where we get the verb. Any questions about that? All right, if you download the PowerPoint, you can watch this in action. It shows glycolysis, and it shows fermentation. Not just the one we talked about, which is the one in the muscles where you produce lactic acid, but also the ones that we're about to talk about, like in yeast that produces alcohol. Every chapter has this, the process of science. I'm always going to skip it because I'm never going to ask you test questions about it. There might be questions on the study guide. I still highly recommend reading it and letting me know if you have questions about it. But again, it won't be on the exam. Come on. I have to finish this up. We've got 6 minutes and some change. Here we go. Fermentation in microorganisms. So we're done talking about fermentation in human muscle cells. Now we're talking about fermentation in microorganisms. Fermentation can sustain many microorganisms. Because think about it. We need a lot of ATP because we're very large, complicated organisms. We need a lot of ATP. That's why fermentation only works a little bit. And then after a while, you die because you don't have enough oxygen. But if you're a single-celled organism, you know, fermentation is just fine. You don't need a lot of ATP. You don't have any muscle cells working. So it's a lot easier. Your book points out that yeast also does make lactic acid. I'm going to put an X to this before I even get into it. So I'm not going to ask you anything about this on the exam. Little side note here. Yeast are really interesting because they are single-celled organisms, but they're also eukaryotes. We are eukaryotes, right? We're the more complicated. So when they go through fermentation, they can either do what we do and produce lactic acid, or if there's not, the conditions are right, they will go through a different type of fermentation and make alcohol. First, we're going to talk about the one that makes lactic acid. You don't need to know this, but what is that lactic acid used for? To make cheese, sour cream, yogurt, soy sauce, pickles, cabbage, olives, sausage meat products. You don't need to know any of that, but basically, again, yeast is really interesting because they can do both. And that's relevant to your everyday life because this lactic acid is used to make all kinds of stuff that you might be used to. Like I already said, yeast is also capable of cellular respiration. I didn't say that yet. I was just talking about fermentation, and it can make either lactic acid or alcohol. But it's also capable, not that you need to know this, I'm not going to ask you any questions about yeast, but if the conditions are right, if there is oxygen, it'll go through cellular respiration. As a matter of fact, that's one of the not the harder things, but one of the requirements when you brew alcohol, like wine or beer or whatever, you have to make sure it's anaerobic. And if it's aerobic, then yeast is just going to be going through regular old cellular respiration and won't be producing alcohol. Anyway, yeast performs alcoholic fermentation, and that's finally where we're getting at here. We've already talked about lactic acids, so let's talk about alcoholic fermentation. You don't need to know about that word. But in the process of doing so, it also produces carbon dioxide. So if you know anything about, like, again, brewing beer, that's where the carbon dioxide comes from, because that is a byproduct of it. Or if you use yeast to make bread, that's why the bread rises, because it literally produces carbon dioxide. Yeah, I'm gonna skip through all this. Here's a picture. This picture should look really familiar. We're taking glucose, we're breaking it down into 2 molecules of pyruvate, just like we did in fermentation of the human muscle cells. We're still stripping away high energy electrons. The high energy electrons still need to put it somewhere. But in this case, what they do is that pyruvic acid, yes, it's going to take those high energy electrons, but it's also, we're losing a carbon from each one, which is where the carbon dioxide comes from. And then we produce ethyl alcohol. All that to say, for the test question, fermentation in microorganisms produces what? And the answer is ethyl alcohol, or even just alcohol. I'm not going to try to trick you. Anything that has to do with alcohol, that's the answer. I'm not going to be like isopropyl alcohol, because that would be wrong, right? It's not isopropyl alcohol, but that also, I'm not going to use that as an option. So again, human muscle cells produce lactic acid, and microorganisms can produce alcohol. It's still basically the same process, right? You're still dumping electrons back on the pyruvate. So any questions about that? All right, your textbook always ends every chapter with the evolution connection. Again, I'm not going to ask you any questions about it. There might be questions about it on the study guide. I still highly recommend reading it and letting me know if you have any questions. If you download this PowerPoint, you can watch this, look at this review. It'll help you study. I'm not going to go through it because you've already seen it, but it's there for your own studying. So the last word for attendance is going to be red, as in Red Friday. Speaking of which, for those of you online, I'm signing off, and for those of you who are here, if you want to do the picture, let's get her done. Everybody have a great weekend.